Thank you all so much for being here. I'm excited to be able to talk to you guys about my favorite subject in the world, which is fungal sex, even though it's been kind of a tough week in many ways. So I'm in Cameron Curry and Anne Pringle's labs here at UW. And to start out, I wanted to define what I mean by sex for most of the talk. And when I switch to a different definition, I'll make that clear. But I'm going to use Graham Bell's definition from his 1982 book, The Masterpiece of Nature, where he defines sex as a process in the course of which genomes are diversified by a type of nuclear division called meiosis, in which the ploidy of the cell is reduced and recombination occurs between sibling chromatids, and that is a form of diversification. And then by a type of nuclear fusion called syngamy. And in syngamy, there's also diversification because you're bringing in a completely new genome from another individual. And sex is thought to have evolved in the last common ancestor of all eukaryotes, and it's conserved in almost all eukaryotes as well, including in the kingdom fungi. And it's thought to be beneficial because it's very good at bringing together beneficial alleles into the same individual genomes. And it's also good at purging basically associations between deleterious and beneficial alleles. So this is kind of a classic figure from John Maynard Smith's 1971 paper, What Use is Sex, where he's modeling the accumulation of beneficial alleles in individual genomes under sexual reproduction and under asexual reproduction. And you can see that it's much faster under sexual reproduction, especially when you have smaller population sizes. And sex is thought to be especially favored in shifting environments. And in fact, it's thought to be so important for eukaryotes that there's this kind of idea that asexual eukaryotic lineages should be doomed to go extinct because they don't have efficient ways of purging basically associations between beneficial or deleterious alleles. So the ultimate effect of sex is that it diversifies genomes and populations, but a lot of the factors kind of affect kind of how this works. So you can think about population diversity on a kind of a spectrum where you have highly recombining populations on one end, or kind of panmictic genome structures, or strictly clonal population structures. And so, for example, how often sex happens can affect where you fall on the spectrum. So not all organisms are obligately sexual reproducing. In fact, it's kind of odd that like a lot of animals are. But you can also think about the kind of sex that you're having affects this. So if you have more outcrossing, then you're going to have a more recombining population structure. And if you have more inbreeding or selfing, you'll have a more clonal population structure. And then even things like population size can affect this as well. And so even if you don't care about sex, which is kind of, I can't completely understand that because I think it's a very interesting process in its own right. If you work on eukaryotic biology, you have to care about sex because it affects population dynamics, phylogenetic tree influence, disease and resistance dynamics, and you can basically insert your interest of choice and sex is going to affect it. And so I said before that sexual reproduction is thought to be especially favored in shifting environments, and one of these is during host-parasite interactions. So this is Hamilton's 1980 Red Queen hypothesis, where he posited that sexual reproduction is thought to be especially favored under parasite-host interactions because parasites are trying to evolve ways around host resistance, and hosts are trying to evolve new resistance to parasites. And so the hypothesis is named after this quote that's said by the Red Queen during Through the Looking Glass, where she says, "It takes all the running you can do to stay in the same place." So basically, host and parasite are constantly evolving or running, but they're actually staying in the same place on the fitness landscape or the same place. And so for the first part of my talk, I'm going to talk to you guys about, I guess, my fungal parasite of choice, which is Ascobopsis. It's an ascomycete fungi in the family Hypocreaceae, and it's a specialized obligate mycoparasite of the fungus-growing ant system. So fungus-growing ants, which are also known as attine ants, are obligately mutualistically associated with a basidiomycete fungus, usually of the genus Leucoagaricus, which they completely rely on for food. And in return, they provide Leucoagaricus with aid in dispersal and also basically protecting it from pathogens and parasites. And one of these parasites is Ascobopsis, which is obligately specialized on Leucoagaricus, so it can't really grow outside of the system. And it also relies on Leucoagaricus for its food. So here we have Ascobopsis plated towards the edge of this plate, glowing towards Leucoagaricus, which it will break down and consume. So in the past 2 years, 2 papers have come out with whole genome-based phylogenies showing that Ascobopsis has co-diversified alongside the fungus-growing ant system, with specific lineages of Ascobopsis being co-adapted to specific fungus-growing ant groups. So the closest living relatives of Ascobopsis are also actually parasites of basidiomycete fungi. So we have Cladobotrium and Hypomyces. And then a little bit farther out, you have trichoderma. And in fact, a lot of trichoderma species are also mycoparasites. So the divergence dating suggests that Ascopopsis probably followed Leucoagaricus into the fungus-growing ant system when it originated 66 million years ago. And you can actually kind of see this when you compare the phylogenies of Ascopopsis and the phylogenies of fungus-growing ants. So there's kind of three main groups of fungus-growing ants, or actually four for this talk. So I apologize for the kind of the teleological naming, but the earliest diverging group of fungus-growing ants are called lower attines, and they practice kind of something called lower agriculture. They tend to have smaller nests. And in fact, the earliest diverging Ascopopsis group is specialized on these as well. And then you have this lineage here of fungus-growing ants, which I actually don't usually talk about, but is important for this talk, which have secondarily switched to growing a different basidiomycete fungus that's in the same order as Leucoagaricus, but not in the same family. It's a coral fungus. And so these Ascopopsis, it's a little bit different here. They actually fall kind of as sister to the higher and the leafcutter. And then one of the more derived groups are the higher attines. They tend to have larger nests. And then this highly derived monophyletic lineage, the leafcutters, we kind of think of them as important because they primarily provision their fungal cultivar with fresh leaf material. And in fact, one of the most highly derived groups of Ascopopsis is specialized on these as well. And I'll say there is an arrow here, and it will be important later, but you can ignore it for now. So given that Ascopopsis is a virulent parasite, you might expect it to undergo sexual reproduction, but actually it has no known sexual cycle or teleomorph, which is what we call kind of the sexual structure of fungi. And when its genome was first sequenced in 2016, and then this was actually also asserted by a new paper that came out on more strains from 2024, people said it didn't have a complete mating type locus. And the mating type locus is the master regulator of sexual reproduction and sexual compatibility in fungi. So if you don't have it, it's kind of like, what's going on? And so this is the figure from the paper where they talk about this. And this right here is where they should have found 3 genes that are in one of the mating type loci, but they actually didn't detect annotation in it with them. If you're noticing, they detected transcription of a gene, which is actually one of the mating type loci, but it didn't get annotated. And at the time, it's kind of like, oh, that's interesting. And that is important for later. But I just decided to kind of ask the question, okay, so does Ascopopsis have a mating type locus? What's happened to it? And going off of kind of this previous information, the hypothesis that I had was that Ascopopsis has lost it, and the chromosomal region that it would have been in will either show evidence of pseudogenation or a complete deletion. And so my initial approach was to take advantage of the fact that in many Ascomycetes, there's conserved synteny or gene content and order around the mating type locus. So I took 25 genomes that had been previously sequenced, assembled, and annotated by another Curry Lab graduate student, Dr. Kirsten Gade. And I blasted the predicted protein sequences, and this is important, against basically protein sequences for the flanking genes and for the mat locus itself from trichoderma. And then I manually checked and classified strains as either having mating type 1, mating type 2, or no detected mating type. And so in Ascomycetes, there's only 2 mating type alleles. And so you have to be different at these alleles in order to be sexually compatible. They're actually technically more accurately called idiomorphs because they're highly diverged from each other. And in Sidereomycetes, which is the group that Ascopopsis belongs to, the MAT1 or the MAT alpha allele contains 3 genes, which we just call MAT111, MAT112, and MAT113. And then the MAT2 or the MAT-A allele contains a single gene, which is called MAT121. And so these strains are haploid, so they should only have one or the other of them. And so I also wanted to look at the region around where the mating type genes should be. So I identified orthologs using MC-scan and then cut out the mating type region using kind of the flanking genes as bookends and then visualized it using MC-scan. And actually, this method was able to successfully identify the mating type locus in 7 out of the 25 genomes that I looked at. So each of these lines represents a genome, and it represents kind of what that region looks like in that genome. And so these 4 genomes here all had the MAT2 locus. They had this MAT121 allele. And these 3 genomes here all had the MAT1 or MAT alpha locus. And I said before that there's 3 genes that should be here, but you can see that we're actually only detecting one, this MAT111 gene. But in fact, the synteny around this region is completely conserved, including in the majority of the genomes that had no detectable mating type locus. And another kind of interesting thing about this is there's not really a pattern as to which ones have it and which ones don't. So this strain here, ICBG 712, is incredibly closely related to ICBG 721. But one of them has the mating type locus and one of them doesn't, and that's kind of unexpected. So I was wondering, is the mating type locus really missing, or are we just not picking it up based on the fact that we're looking at the predicted protein sequences? And so what I did is I took the 25 genomes and then I took an additional 14 from this 2024 paper, which I didn't include in the Synteny analysis because they didn't upload any like protein or GFF files, which I kind of needed for MC scan. And I did a T-BLAST X with the protein sequences of all 4 mating type genes from Escovopsis when we had them, or from Trichoderma for MAT112 and MAT113 because we didn't have those genes. And I manually checked to see if there were hits that were in the right syntenic region. And then I actually ended up having to manually annotate the mating type locus in all 39 genomes, which I did in IGV. And it's kind of hard to explain what this looks like, but IGV is a genome visualizer. So you can load in your genome and you can load in any tracks. In this case, there's no models in the GFF file. And I just used the protein sequences from the outgroups or from Escovopsis when we had them to guide this annotation. And it turns out that actually every single Escovopsis strain had a complete mating type locus, including MAT112 and MAT113 in the MAT1 strains. And this was not surprising to one of my committee members who was like, I know that they're there. So I'm not really going to get into why this happened. I assume that something is happening where these models are being called and then being culled in kind of the trimming process in the protein annotation. I spoke to several people at the Mycological Society of America conference, which is like the major conference for mycologists, who were like, I've had this problem. Like, we have a known sexual cycle. We know that the MAT loci are there. They're just not getting called. And I think this is probably partially, especially because we are not modeling it fungi super well, and especially these lineage specific genes, MAT112 and MAT113. So if people want to talk about that, we can, but I'm not going to mention it again. So next, I wanted to understand if the mating type loci showed signs of conservation and functionality. So the gene structure for all 4 genes is highly conserved, including exon and intron number, exon and intron length, splice site location, and sequence, and there's no premature stop codons in any of the genes. And I also validated my manual annotation of MAT111, which was more accurate than the predicted protein 1, using the previously generated transcriptome from the 2016 paper, because we actually had what it was going to look like. And so next I looked at conservation of the functional domains. And so I'm only going to show you MAT111 and MAT121 for time's sake, but these results hold for MAT112 and MAT113. So MAT111 is this alpha box transcription factor. And here I've kind of pulled out the most conserved part of that functional domain, and highlighted in purple are the most important sequences, the most highly conserved sequences. And you can see that in all of our Escovopsis that have MAT111 or the MAT1 allele, both the placement and the sequence of this domain is highly conserved. And then MAT121, so these are different strains because strains either had one or the other. There was one exception. I think it's a mixed culture. I won't get super into it. But MAT121 is a high mobility group transcription factor. There's actually really good modeling for this. So we know that this W that's in bold is important for pairing with the DNA backbone to promote transcription. And it's completely conserved in Escovopsis as well. So I'm kind of, in my mind, looking at this, it makes me think that these genes probably are functional. So next, for each individual gene, I determined the strength and type of selection that they were under. And I used this program called Fast Unconstrained Bayesian Approximation through the HiFi package to do this. And basically what it does is it looks at the rate of synonymous mutations, which we'll call beta, to non-synonymous mutations, which we'll call beta, to synonymous mutations, which we'll call alpha. Because under purifying selection, you should see a much higher rate of synonymous mutations to non-synonymous mutations. And so on the left, I'm showing you 2 plots for MAT121. And this plot is showing the strength of purifying selection, where you're basically looking at beta minus alpha. So the more negative it is, the stronger the purifying selection is. And you can see that there's a strong peak around -3, which indicates that a lot of the gene is under probably strong purifying selection. And over here, I'm modeling the posterior probability that each codon site in the gene is under purifying selection. And 0.9 is kind of the it probably is, and then 0.95 is that it's really probably under purifying selection. And so this dotted line here is at 0.95, and you can see that the majority of codons are probably under purifying selection in this gene. And so this pattern also holds for the MAT1 locus. So here is for MAT111. You can see again that the strength is centered kind of around -2, it's not as strong. And then the density is also centered at most sites are probably under purifying selection. It's definitely, actually, I would say it's more prominent for MAT 112. And then it's the least prominent for MAT 113. You can kind of see that there's multiple density peaks and that a lot of these sites were under super strong purifying selection, where, it's not cutting, the density plot is kind of centered not in the peak is around 1, but it's not like it doesn't start before 0.95. So next I wanted to model the evolutionary history of the mating type loci. But before I show you the results, I'm going to go back to the evolutionary history of Escovopsis overall and tell you why I had that arrow in the first slide. So earlier I only showed you this phylogeny on the left, which is a maximum likelihood concatenation species tree. And if you use this approach, then you get this topology S, where you have a single origin of association for the coral Escovopsis. So they kind of all group together. But if you use a maximum likelihood coalescent approach or a Bayesian approach, you get this topology, where actually this lineage of coral Escovopsis groups with this other newer lineage of lower Escovopsis and not closer to the other coral associated Escovopsis. And I'm going to call this topology M for multiple origins of coral associated. And if you look at the single Quapi orthologs for this lineage, about half of them have topology M and about half of them have topology S. Like it's a pretty even split. Okay, so now what's going on with the MAT locus? So to reconstruct the phylogenetic relationships for each mating type locus, I used the maximum likelihood program RAXML with transfer bootstraps. And I actually also reconstructed the phylogenetic relationships for the flanking genes because I wanted to see what was kind of going on in this region. And so for MAT 121, which I'm going to talk about first, this phylogeny is only based on basically strains that had the MAT2 allele. But these are actually based on all 39 strains because when I tried doing them separately or together, it was just completely all the same. So I was like, might as well just put all of them into the big phylogeny. And so this actually has topology M, which is when you have multiple origins of association. And really interestingly, we see an introgression of one of our non-ant-associated Cladobotrium species between this lineage and the rest of the Escovopsis strains for MAT 121. And actually, if you look at this COX-13 gene, which is just one of the flanking genes, it also shows this like topology M with the introgression of this Cladobotrium strain as well. And so I was kind of interested, notably I'll say, so MAT 121 is kind of closer to this APN2 gene. And so this side, a lot of the genes have topology M. And then on this side, you have topology S. And then when you get out to this gene, it also has topology S, which we can talk about maybe why in a few minutes. It might just be that you kind of, this region is going to be under reduced recombination, but as you walk out, you're going to have more recombination events happening. So I also looked at the 3 MAT1 genes, and I did each of these separately. And so here we have MAT 113, which has topology S, so it has a single origin of association with a coral. But MAT 112 and MAT 111 actually have topology M with the multiple origins of association. And this was very surprising to me. I kind of thought they would all have the same topology. And even more interestingly, I expected MAT 111 to have topology S because it's close to this gene SLA2, which also has topology S. But it's actually kind of the reverse of what you would expect, where the genes on this side of the MAT1 locus have topology M and the genes on this side have topology S. So I did test for recombination within each individual gene using a package in HiFi called GUARD, and there wasn't any evidence of that. I mean, clearly there's recombination between the genes because they have different topologies, but there's nothing like within each gene that I think is driving this. So my next thought was, okay, maybe there's an inversion that's happened sometime in the past in the MAT1 locus specifically. And so I used MC-scan again to basically examine the synteny around the MAT loci and the outgroups. So here we have Escovopsis strains for MAT1 and also for MAT2. And then we have our Cladobotrium and Hypomyces, which are the most closely related. And then you kind of walk out, we have Trichoderma, which is in Hypocreatiae. And then, unfortunately, the closest related genome that had like a good assembly was in Hypocreales for cordyceps. And I apologize that this color is not showing up super well on the screen. And so when you look at the Escovopsis and the Cladobotrium for both MAT1 and MAT2, they kind of have the same arrangement here. But actually, there's an inversion in both of the Trichoderma strains, which I kind of chose because they're there's two, branches of trichoderma, and they're in both of them. And so you see that both for MAT1 and for MAT2. But then when you look at cordyceps, which is our hypocreales outgroup, it doesn't look like trichoderma. It looks like the Cladibacterium and Hyphomyces. So there's kind of two possibilities here. The first is that this inversion at the mating type locus happened in the last common ancestor of all trichoderma. And the second is that this inversion happened in the last common ancestor of Trichoderma, Cladibacterium, Hyphomycetes, and Escovopsis. But then there was another inversion in Hyphomyces and Cladibacterium and Escovopsis. And it's more parsimonious that there was only a single inversion and not that there was an inversion and another inversion. But the problem is that we don't have a lot of good genomes representation for the Hypocreaceae, and we would probably want more representatives if we were to figure this out. But it is kind of interesting if you think about it, because this might make a little bit more sense why these genes, which were all involved in the inversion, are all the ones that have topology M. And SLA2 and CIA30, which are not involved in the inversion, have topology S. Don't 100% obviously have an answer to why this is, but it is pretty suggestive that there is some kind of ancient hybridization event in my mind, the fact that we have this really 50-50 split and that it impacts the mating type locus. So in summary for this project, the mating type loci and Escovopsis are present and they're conserved, suggesting that Escovopsis is probably able to undergo sexual reproduction. Even though the MAT loci were not in the predicted protein sequences, they are present in the genome. And I think for me, this is kind of a lesson that you can never take evidence of absence for evidence of absence. You always need to go and look and make sure that the reason that you're not picking something up is because it's not there and not just because maybe it's not being called in your predicted protein sequences. And in addition, the MAT loci are all under strong purifying selection. The whole genome phylogeny and then the MAT phylogenies show discordance in placement for one lineage of coral-associated Escovopsis strains. So this lineage here. And so kind of what I'm working on now, and it's funny to go after Claudia's talk last week, but I am working on using SNAC, Phylodiamond, and Phylonet to test if and between what strains hybridization is occurring. And I'm working on getting the bootstraps for that right now, but I will say that the most likely number of hybridization events for SNAC was 2. So I do think something is going on, probably from a ghost lineage, but we'll see. Okay, so now I'm going to switch to telling you about a different project, which is also aimed at understanding sex in fungi. And this is a theoretical project and not a data-driven project that I've been working on with one of my advisors, Anne Pringle. And so the first part of the talk, I was talking about ascomycete fungi. And now I'm going to switch to talking about basidiomycete fungi, which are another phylum of fungi. They contain the charismatic mushrooms that you think of, but they also contain smuts and rusts and cryptococcus for people who are into cryptococcus, which is cool. And so I kind of was wondering, from a quick show of hands, if people in the room have heard this claim that some fungi or one fungus in specific has 23,000 sexes or mating types. And okay, so some of you have heard it from me, but some of you I know haven't heard it from me. So we'll say that, it has been making the rounds, not just because of me. And so Anne and I noticed this claim popping up in a lot of popular science books, including An Entangled Life by Merlin Sheldrake, which was hugely popular. It was on the bestseller list, and they issued a reprint. And he writes that some fungi have tens of thousands of mating types, approximately equivalent to our sexes. The record holder is the split-gill fungus Schizophilum commune, which has more than 23,000 mating types. It was also in this book, which was published this year. This is a book for people who love mushrooms, is the name of it. And she writes, the most astounding and difficult to grasp reproductive adaptation of Schizophilum commune is that they have as many as 28,000 different sexes or distinctive mating types. I actually don't know where she got 28,000 from. I've never seen this number before in any of the literature. I think it was probably just a typo or something. It is 23,000. That is the claim that we're mostly working with. But and then it was also an NPR, which said that Schizophilum commune, or the split-gill mushroom, has 23,000 sexes, making it somewhat of a queer icon in the field of mycology. And so in all these places, there was no accompanying citation or explanation for where this information was coming from or based on. And me and Anne were really interested, what is the history behind this claim? What are the studies that this is based on? And this here is actually, this is a picture of Schizophilum commune, the split-gill mushroom. It's very beautiful. It makes bastiocarps or mushrooms on decaying wood. And it's also kind of fuzzy, and I think it's just gorgeous. But Anne and I were interested in 2 specific questions. So one is this claim true? Does Schizophilum commune really have 23,000 sexes or mating types? And what is the like science behind this? And the second is all these places are saying that mating types and sexes are equivalent to each other or roughly equivalent to each other. Is that really true? And should we be saying that? And so in order to understand where this claim comes from, we have to first understand the biology of Schizophilum commune and Basidiomycete fungi in general. And what better way to do that than with a life cycle? So this is a life cycle made by Sarah Friedrich in the botany department, which is a part of me and Anne's paper on this, which is currently in review. And so mushrooms are the sexual structures of Basidiomycete fungi, of most Basidiomycete fungi. Some of them actually don't make mushrooms. So apologies to that, because I know there's someone in here who works on Cryptococcus. They're the sexual structure for a lot of Basidiomycete fungi, and they are where the gametes are formed, which in this case are spores. And so the spores contain a single haploid genome. Most spores have a single nucleus in which that haploid genome is contained, but actually Schizophilum commune is kind of weird. It has two identical nuclei in its spores, which contain the same haploid genome type. So these spores germinate to form monokaryotic hyphae. And if two monokaryotic hyphae meet each other, they will fuse. And if they're compatible with each other, they'll undergo something called reciprocal nuclear exchange. And compatibility in this case is controlled by the mating type genes, which really you can think about as genes that control the formation of a new dikaryon from two monokaryons. And in the case of most Basidiomycete fungi, there are two unlinked mating type loci, which we're going to call A and B. This is something known as tetrapolarity, which I'll touch on in one more slide. But importantly, this compatibility basically is, are you identical at the mating type loci? If you are, you're not compatible. If you're different at both mating type loci, you are compatible. And so you'll undergo reciprocal nuclear exchange, which is, in my mind, kind of my favorite part of the Basidiomycete life cycle, where nuclei from one side of the monokaryon migrate and divide into the other side. And the same thing happens from this monokaryon as well. And what you end up with is that these nuclei don't fuse with each other. You get this dikaryotic arrangement where every cell contains two haploid nuclei that are genetically distinct from each other. Kind of amazing if you think about it, because this really is different from the way that we think about sex in a lot of organisms. It's easiest to kind of compare it to humans in the sense of like sperm and egg cells, which are gametes, fuse, and then the nuclei immediately fuse with each other. The fusion between these nuclei actually only happens in specialized cells in Basidiomycetes. So the dikaryon will grow for days, weeks, months, years in whatever substrate it's growing in. We don't 100% know how long dikaryons last, but under this the right environmental conditions, they'll make mushrooms or the sexual structures. And one dikaryon can and often will make multiple mushrooms, both in the same season and if it lives longer than just like one growing season. And in the spore-producing cells of the mushroom, the nuclei do finally fuse. So there'll be a diploid nucleus very briefly before they immediately undergo meiosis to form four recombinant nuclei. And then actually in most mushroom-forming Basidiomycetes, there's another round of mitosis, and you again get these kind of dinucleate spores, which will germinate, and the cycle starts over again. So I said that this arrangement of having two unlinked loci is known as tetrapolarity, and the reason for this is that, so if you have like a dikaryon, it will have a nucleus that will say have one allele at A1, and another allele and one allele at B, B1, and then the other nucleus will have different alleles at those two, so A2, B2. And so there's four possible spores, like there's four possible mating type kind of combinations that are possible in the spores. So you'll have A1, B1, A2, B2, those look like the parents, but then you'll also get A1, B2, and A2, B1. And so these spores, each individual kind of type of spore is only compatible with 1/4 of the other spores produced from that mushroom, because you have to differ at both of those. So A1, B1 is only compatible with A2, B2, and then the same can be said for A2, B1. So the A mating type locus encodes homeodomain containing transcription factors, and there's usually two main genes, HD1 and HD2. These are actually need to form a heterodimer with each other in order to promote transcription. And HD1 and HD2 in a single genome are not compatible with each other. So HD1 cannot fuse with this HD2, it has to fuse with the HD2 from another genome. And the B mating type locus contains pheromones and pheromone receptors. And the same thing is true again, where pheromones and pheromone receptors in a single genome are not compatible with each other. But the situation is actually even more complicated in Schizophilum commune, and this is part of the reason that there's so many mating types. So each of the loci are further subdivided into two subloci. So we call one of them A alpha and we call the other one A beta. And A beta contains multiple copies of both the HD1 and the HD2 genes. And again, as I said before, none of these copies are intercompatible with each other. And then the B locus also is subdivided into B alpha and B beta. Again, none of these are compatible with each other. And so we'll call the sum of the alleles at A alpha the A alpha mating type, and we'll call the sum of the alleles at A beta the A beta mating type. And then the sum of all of those alleles together is the A mating type. And the same thing is also true for the B mating type locus. So this is just kind of like amazing if you think about it. There's so many ways that you can think about different alleles forming, and that's partially probably why there are so many different mating types. Okay, so the estimates that Schizophilum commune has 23,000 mating types mainly derive from a single study and then a couple of follow-ups called the number and distribution of incompatibility factors in Schizophilum from John Raper, Gladys Kronglev, and Marjorie Baxter. And so what they did is that they collected and were sent basically mushrooms of Schizophilum commune from all around the world. So they actually did have samples from every single continent except Antarctica. But the majority of their samples came from Massachusetts because John Raper was at Harvard, and actually also from Madison because John's brother Kenneth Raper, who if anyone has like ever been to the Kenneth Raper Symposium, was at Madison. And so 2/3 of the sample are actually kind of from the northeast central United States. And so why did they care about this? Well, the diversity of alleles, how many alleles there are, controls how many germinating spores can form basically dikaryones with each other. And then this shapes population dynamics and ultimately evolution. And if you have something like thousands of mating types, you're probably going to be compatible with spores from every other mushroom except the ones from your own in the system. And so this is like really important for thinking about how sex works when you have things that you're germinating and you can't like really move anywhere. Okay, so they took advantage of the fact basically to count how many A and B alleles. This was all done before DNA sequencing, and I actually think this study is really beautiful. But they ended up deriving 2 monokaryons from all these, all the mushrooms that contained all 4 mating type alleles from the parents. So one that had A1B1 and then one that had HB2, and they ended up with 114 monokaryotic strains. And they paired all of these monokaryons with each other. And I think it was something like 5,000 pairings, like a large amount. And they were able to tell basically if differences were, or incompatibility was coming because of they were identical at A or B, because you get slightly different reactions because the genes control slightly different things. So if you are different at the A locus, but identical at the B locus, what happens is you get this thing. So in Basidiomycetes, including Schizophilum, a lot of them have things called clamp cells. These are little kind of like nub cells that form between 2 hyphal compartments, and this makes sure that every cell contains 2 nuclei in them. But if you are identical at the B, you can form this clamp cell, but it can't fuse back with this cell here. And so this nucleus just becomes trapped. But if you're identical at A and different at B, you can't even form clamp cells because the A is responsible for the formation of clamp cells. And so you'll have kind of septal dissolution in the nuclei and will migrate, but you'll end up with this kind of nubby morphology, this like giant multinucleate cell. If you're identical at both, you just keep growing as a monokaryon. And if you're different at both, you will form a dikaryon. So they were able to kind of look at all 4 types of these reactions to say, okay, like which ones share which kinds of alleles. So they ended up counting 96 unique A alleles and 56 unique B alleles. And then they did some modeling to say basically if we kept sampling, how many more alleles would we find? And they ended up estimating that they would have 339 A alleles, although like, you know, with between 217 to 562, so kind of a big range, and 64 B alleles. And here's where the like kind of an additional step of logic comes in. Each mating type is the unique combination of A and B alleles. So if you multiply the number of possible, like the total number of A alleles by the total number of B alleles, you'll get the total number of possible mating types. And in this case, it's 21,696. So we're not quite at 23,000 yet, because they weren't able to distinguish between basically differences that were derived from either the subloci. So they were only able to look kind of like at A and B as a whole. So then there was some additional work that was on all these 114 strains. And what they did was basically they were able to link mutations to either the A alpha or the A beta. And then they did this also with the B loci as well to tell where the differences were coming from. And they counted 9 A alpha and 25 A beta alleles. And then they did further modeling again to say, okay, there's probably 9 A alpha alleles and 32 A beta alleles. And then they also did this with the B alleles. So they found 9 B alpha and 9 B beta. And then I don't actually fully, I've tried to understand this a lot. They looked at basically the recombination rates between these B alleles and were like, these are actually probably the total number that are found in nature. And so what you do then to get the total number of mating types is you multiply all these by each other, because the total number of A alleles is the total combination between basically A alpha and A beta, and then same for B alpha and B beta, and then you want to multiply those all together. So if you times 9 by 32 by 9 times 9, you get 23,328 possible mating types. But if you want to be more conservative and just go with the number that they actually observed, you get 18,225 possible mating types, but a lot of mating types. So there's kind of 3 key assumptions that go into like, are there actually 23,000 mating types? And at the time that they did this, they had reason to believe that all 3 of these should be true. So the first one is the most important. It's that all 114 strains belong to a single species with limited geographical population structure. And they believed that this was true because all of the strains were basically like, no matter if they found them in Australia or if they found them in Massachusetts, they were able to form dicaryons as long as they differed up the mating type alleles. And they found shared mating type alleles between strains that were on different continents. And it didn't really seem like there was any structure to like the number that were shared versus the number that were unique. So they were like, okay, I think that this is probably true. And then based on this, you get that the idea that basically all combinations of alleles are possible in all populations. And then also that the mathematical modeling is accurately predicting the number of missing alleles. And so I'm going to start by looking at this first assumption because it's actually the most important. And we actually know now that it's not true. So there's been a couple of studies that have done basically DNA sequencing, and we know that there's 2 to 3 distinct independently evolving populations of Schizophilum commune that have very limited gene flow between them. So there's one lineage in North and Central America, there's one in South America and the Caribbean, and then there's one in like the Eastern Hemisphere in Europe and Asia. So these species have not been named or delineated. We call them a cryptic species complex still. And you might be wondering, okay, so there's 2 to 3 species, then why are they still all intercompatible with each other? And why are they sharing mating type alleles across probably long evolutionary distance? And this is because the mating type alleles in basidiomycetes are known to be under something called frequency-dependent selection. So if you think about it, if you have, and I'm just using spores and skipping the monokaryons. So if you have one spore that has a rare mating type allele, it's going to be able to form dicaryons with every other basically monokaryon in the population. And so it's going to be more represented in the next generation than it was in the first. And so basically any allele that's rare has a frequency dependent advantage under this. And you can also talk about negative frequency dependent selection, but in this case it's positive. And so you also have very large population sizes, and both of these buffer against the loss of mating type alleles, and are probably what's causing this phenomenon. So this second assumption is based on the first. This is the logic behind the multiplication. It's that all combinations of alleles are possible in all populations. And without delineating species boundaries and going back to count, we don't know how many alleles are actually species specific. And we're also probably missing alleles from undersampled populations, which is really just anything outside of Northeastern and Central USA. So certainly from like South America and like the Eurasian populations. And then the third is that mathematical modeling is accurately predicting the number of alleles. But actually, and I mean, they would tell you this too, like I think most people who use mathematical modeling, it's our best guess. And these models also rely on assumptions from 1 and 2. And so in conclusion, are there 23,000 mating types? The answer to question 1, probably not. Until a mycologist delineates species boundaries and goes back to count what alleles are species specific, the extrapolated estimates of having 23,000 mating types are numbers of mating types have to be understood as hypotheses. What you can take away from this is, yeah, there are thousands of mating types. Is it 23,000? Maybe that's like not the most accurate answer. Okay, but then this kind of brings me to my second question, and this was part of what was so interesting to me is all these places are saying sexes and mating types are equivalent. And now I'm going to be moving from the definition of sex as a meiotic process to the definition of sex as a category. And so I first want to start by trying to define what a sex category is. But it turns out it's actually really hard to do that And there's currently an intense discussion in the animal world and in evolutionary biology more generally from a lot of really great thinkers that are kind of wondering, is sex a coherent and useful term? And the best way that I have to kind of demonstrate this is this really great graphic from this historian of science, Beans Veloci, where they asked their students to tell them, okay, what defines different sex categories? And they came up with so many different answers. A lot of these correlate, they may not correlate with each other. And basically the idea is that sex categories really are capturing like a large amount of diversity that's hard to reduce to just one thing. And so I'm going to try to sidestep this debate, which I know is kind of like a cop-out, by simply comparing the ways in which sex is operationalized to mating types and seeing if there's kind of overlap in the way that these categories are used. And so the first category that I identify is, so the definition of mating types is it refers to classes of intercompatible gametes. And sexes can and often do refer to classes of intercompatible gametes. So sperm are not compatible with sperm, eggs are not compatible with eggs if you're kind of thinking about a gametic definition of sex. But there's actually other factors that can control if gametes can fuse in populations, like cytoplasmic incompatibility or genetic mechanisms too. So it's not true to just say that sexes are just classes of intercompatible gametes. So the second thing is that mating types are associated with reduced regions of recombination, and sexes can also be used in a way that are associated with reduced regions of recombination. So if you think about sex as kind of defining it at the genetic level, actually there's a lot of different, like there's a large variation in the region that is under reduced recombination. So in pufferfish, and then also in fact in Schizophilum commune for the mating types, there's actually a very small region that's under this reduced recombination. And you can see the whole spectrum here, where you get all the way up to fruit flies, where there's no recombination between what we call these sex chromosomes. And in fact, the chromosomes that carry the mating types in basidiomycetes can also have very large regions of reduced recombination. So this is the mating type chromosome for Microbotrium, which is an anther-smut basidiomycete. Doesn't make a mushroom, but it is a basidiomycete. And basically the dark gray is the region that is under restricted recombination. It's almost the whole chromosome. But I will say this is actually kind of rare in basidiomycetes, especially in mushroom-forming basidiomycetes. You usually see very small regions of reduced recombination, especially because there's such allelic diversity. So the next way, and I think this is the way that most people think about sex, is as referring to differences in gamete size. So the macrogamete is what we'll usually call the female, and then the microgamete is what we usually call the male. And really importantly, though, mating types are not associated with any differences in gamete size. The spores between that have different mating types are exactly the same. And I think this is where we start to really see a breakdown in the way that these two terms are used. So the next way, oh, sorry. Okay, okay, sorry about that. So the next way basically that people think about, and this is again kind of using that genetic, I'm sorry we're moving through so many definitions of sex. It's really hard to like have a coherent definition. I think that also speaks to kind of the problem with sex as a category in itself. But so mating types are determined at the haploid level. It's the combination of alleles in a haploid genotype. But if sex is genetically determined, and I want to be clear, it often is not genetically determined. It is often environmentally determined or plastic. But if it is genetically determined, it's determined at the diploid level. So for example, you look at the combinations of two different chromosomes, which we'll usually call XY or ZW to say, okay, what is the genetic sex here? And I think this is a really important difference, the haploid versus diploid, because what's really doing is describing two divergent mechanisms for how identity of some kind is being determined. So mating types sometimes control the inheritance of cytoplasmic elements, not actually very often in basidiomycetes especially. So the most like well-known example of this in basidiomycetes is Cryptococcus neoformans. So the mitochondrial inheritance is determined by the A mating type, but not in Schizophilum commune, where actually the mitochondria don't migrate, and they don't migrate in most filamentous basidiomycetes either. And so each side of the dikaryon actually inherits its mitochondria from whichever spore parent like it first derived from. And so you'll end up getting like unibiparental inheritance, where each mushroom will pass on a single mitochondrial type. But because mushrooms are going to pop up from different parts of the dikaryon, you'll end up passing on both mitochondrial types. And in sex categories, they're often associated with basically control of cytoplasmic elements. So, you know, if we think about a lot of animal sexes, the macrogamete is the one that usually contributes to mitochondria. That's not always true, though, and there can also be leakage. And then I think this is also, I mean, I think these are all important, but I think this is also kind of a really important one, which is that the operational unit under discussion in mating types and sexes is different. So when you're talking about mating types, what you're really talking about is the nucleus or sometimes the monokaryon, but you're often talking about the nucleus because when you talk about the dikaryon, people end up talking about like the nuclei as like determining the mating types. And when you talk about sexes, the operational unit that's under discussion is usually the whole organism or sometimes the gamete itself. And so there's like a really stark difference here that kind of relates to the diploid haploid level. And part of this too comes out to play in how these are functioning during the life cycle. So mating types are actually critically important for maintaining the dikaryon as it grows. And I don't really have a good analogy for that in like humans or animals because it's just such a divergent way of living. There's not really a good comparison. So there's no physiological differences or extended phenotypes between basically monokaryon spores or dikaryons that have different mating types, but sexes are often associated with basically extended phenotypes or physiological differences between bodies that we call different sexes. So an example of this would be ornamentation or hormonal differences. That's not at play at all with the mating types. So this is also important, perhaps most of interest to this class, which is that they probably evolved for different reasons. So mating types likely evolved to basically promote outbreeding and reduce inbreeding. And this is actually why you see such a large number of mating types probably in the basidiomycete fungi as well. But our understanding of the way specifically different size gametes evolved is that they're probably being shaped by disruptive selection, where there's selection for smaller gametes that are more mobile and greater in number, and then there's selection for a larger gamete that perhaps contributes more to the zygote. And then I actually think this is really important for scientists to think about too. There's like no cultural baggage for mating types. I wouldn't say I've really met a mycologist that has an opinion on specific mating types before, but there's a lot of cultural baggage with sexes. And in fact, people use that to import ideas that are discriminatory into science or even shape their science around these things. So you might be wondering, okay, you've detailed all these like ways that mating types and sexes are not the same. Why are you being so pedantic about this? Like maybe even if they're not really the same, it doesn't matter. Maybe it's still a good science communication tool, but I actually believe it matters a lot. And I'm just going to read you this quote from me and Anne's paper, where we write, animal sexes and fungal mating types are not synonymous with each other. And perhaps especially because of an emerging understanding of the fractured meanings of sex in animals should not be used interchangeably. How wondrous it is that reproduction can be so different between and within two kingdoms of life. So I think that equating them pushes this very confusing metaphor that obscures the biology of fungi and does a disservice to people when we're teaching them about fungi. But also, I mean, really, mating types are describing a very specific thing. And sex categories are, as we know now, describing multiple fractured things that may be associated with each other. And so it aligns with the discussions that are happening about sex as a category in general to say maybe we should just walk away from using it as a catch-all and use a term that we're defining in a specific way, that we understand in a specific way in the biology of our organism. And then I'm just going to read a statement that I wrote when I woke up on Wednesday really quickly because it's really important to me. So I woke up on Wednesday morning and I felt a lot of deep grief and fear, especially for my trans friends and families and loved ones in community. And I want to say that I spent a whole lecture talking about sex, and it would feel wrong to not address what I view as a deeply interconnected issue, which is that politicians will take and distort and twist science, and sometimes they don't even need to because it's already baked in with bias, to further an agenda to hurt and exterminate trans people. And if you're a scientist that studies sex or talks about sex, or it's even a factor in your work, you have a chance to do better and to change how you formulate or think about or phrase questions to make it harder and to refute this pernicious and hateful phenomenon. And I know it's ironic almost to say this after I spent so much time telling you that mating types and sexes are not equivalent. But honestly, I feel like this is how I can help my field because I know and I have seen how when people use these interchangeably or import sex categories into fungi, it opens the door to importing sexist and transphobic language or metaphors. And I don't want that because it contributes to a worldview that not only do I think is biologically wrong, but as Myrna Perez said, I don't want to live in that world. Anyway, this is obviously one small thing in the scheme of everything, but I think it's the most I can say or articulate about this right now. And honestly, if you take one thing away from my talk, I hope that it's that you should question the way that you're using sex or categories like that in your research and the way that it can be mobilized for political reasons. And I just, want to acknowledge both the Pringle Lab and the Curry Lab, which I love all of my lab mates there and the support that I've gotten from Anne and Cameron, as well as people from the Feminist Science Reading Group and my thesis committee members and funding sources. And with that, I'll take any questions about any of the projects. So.